This is Biology 101, Chapter 13, Meiosis. Okay, if you remember back to the last chapter that we discussed, in Chapter 12 we were talking about mitosis. And mitosis is the type of nuclear division that we see in somatic cells. Remember, somatic cells are going to be the cells that make up most of your body. However, meiosis is the type of nuclear division that we see in gamete cells. And gamete cells are the reproductive cells that are passed from parent to offspring to create a zygote. And that zygote is going to then go through the process of mitosis and cell division in order to reproduce to create a bunch of cells that will eventually become a fully formed organism. And so it's very useful at this point because we're looking at how those gametes get made and the role that that plays in inheritance to kind of take a little introduction to genetics and take a look at that. So when we're looking at genetics, we're looking at the scientific study of heredity or inheritance. And inheritance is really just a look at how traits get passed from parent to offspring. And if you look at your siblings, for example, you may notice that within a family, your siblings are all going to be different. They're all going to look different. So there's variation among siblings, unless, of course, you have identical twins, which is a completely different situation. But there's variation or differences among siblings. However, you do see many similarities between those siblings, and then also similarities when you look at your siblings, yourself, and your parents. And so the reason for that is because when parents pass traits on to their offspring, they don't pass all of their traits on. They're only going to be passing on one gene for every trait rather than the two that they actually have, which means that every offspring is going to get a completely different combination of genes from mom and a completely different combination of genes from dad. And the way that these combinations of genes are made is through the process of meiosis. And so continuing our discussion of genetics and heredity, one of the things that we need to understand when we talk about genes are really just pieces of DNA, or relatively short pieces of DNA that are found along the length of a chromosome. And genes code for proteins, and the proteins are going to be the things that actually produce that trait. When we look at the long pieces of DNA, remember they get organized, wrapped around proteins, and then wrapped some more. And that organization is what produces the chromosomes that we're able to visualize when we look at the DNA in a cell. And all of those chromosomes are housed in the nucleus of the cell. One of the tools that we can use to look at the DNA in the nucleus is called a karyotype. Now, karyotype is really just a map or a chart of the chromosomes in an individual, and it's very useful in medical genetics because it allows you to look broadly at all the chromosomes to see if there are any abnormalities. And so what we do is we take a picture of all the chromosomes once they've condensed, and we organize them into pairs. And so you can see that we have 2 of chromosome number 1, 2 of chromosome number 2, 2 of chromosome number 3, and so on. So this karyotype is from a diploid cell, and remember, diploid means that it has 2 sets of chromosomes or 2 copies of every chromosome. So if you were to take 1 of chromosome 1, 1 of chromosome 2, 1 of chromosome 3, that would be 1 set of chromosomes. The only chromosomes that are not found in pairs are the sex chromosomes, okay, the X and the Y. In humans, if you have 2 X's, then you would be a female. If you have an X and a Y, then you would be a male. Also in humans, we have a total of 23 pairs of chromosomes, which gives us a grand total of 46 chromosomes in our somatic cells. Whenever we produce gametes, those are going to be haploid, which means they're only going to have 1 set of chromosomes, so you would have a total of 23 chromosomes in every gamete cell. I'd just like to very quickly show this slide because I think it's interesting. Very often we think that as humans we are the most complex organisms and the most intelligent organisms, so we must have the most chromosomes, and it's just simply not true. The number of chromosomes that we find in cells tends to be based on how those organisms have evolved. And so if you look, for example, at chickens, they have 78 chromosomes in a diploid cell compared to our 46. And we know that chickens are not necessarily any more complex or intelligent than we are. However, if you look at chickens compared to turkeys, 78 and 80, They're both birds. They're both poultry, if we think about it in that terms, or those terms, and they have very similar number of chromosomes. Likewise, if you look at humans and gorillas, we're both considered primates, so we're very closely related, and we have similar numbers of chromosomes as well. And so this first column is showing you the diploid number, so this would be 2 sets of chromosomes. And the second column is showing you the haploid number, 1 set of chromosomes. And you can see that all the haploid numbers are exactly half of the diploid number. Excuse me. And that's because that would be the number of chromosomes in 1 set. Okay, so let's take a look quickly at chromosomes, because we are going to be looking at some new terms possibly as we go through our discussion of meiosis. When we look at chromosomes in a somatic cell, they are found in pairs. Okay, and what we mean by a pair is that those 2 chromosomes contain the same type of information. So if we look at this pair in the cartoon drawing below, this would be considered a homologous pair. So maybe this is chromosome number 1 and chromosome number 1. So if we look at them, possibly right here on this chromosome, on every single one of these chromosomes, you're going to have the same type of gene, possibly a gene for hair color or eye color or attached or unattached earlobes. You're going to have the same type of gene there, but possibly on this chromosome you have the gene for attached earlobes, and on this chromosome you have the gene for unattached earlobes. And so they're going to contain the same type of information, but maybe not the exact same information. However, when we look at one of these chromosomes, if you notice, it forms an X. And the reason that it forms that X is because this chromosome has been replicated. If you recall from our discussion of mitosis, we said that in interphase, before the cell actually goes through the process of nuclear division, in S phase it's going to go through process of DNA replication. And in that replication, one of these chromosomes makes an exact copy of itself. So this chromosome makes an exact copy of itself, which we see here. As long as these 2 copies of the chromosome are attached to each other at the centromere, they are considered a single chromosome. But each copy is called a sister chromatid. So this is one sister chromatid, and this is another sister chromatid. Likewise, on this chromosome, it has been replicated. So this is one sister chromatid, and this is another sister chromatid that make up one entire chromosome. When those 2 sister chromatids separate from each other, they become separate chromosomes. Okay. The reason that we have 2 copies of every chromosome in our cells is because we received one from mom and one from dad. So in one of your somatic cells, you're going to have 2 copies of chromosome number 1. And one of those copies came from mom and one of those copies came from dad. And remember, they don't carry identical information in the homologous pair, but they carry the same type of information. Sister chromatids, because they are exact copies of each other, will contain identical information. So this copy and this copy will be identical. Okay, very quickly we need to mention that there are 2 types of reproduction that can occur. One of those is asexual reproduction, in which you only need one parent. Okay, and that one parent makes a copy of itself either through a cellular reproduction event, so single-celled organisms just make a copy of themselves and divide, and now you have another whole organism. Or, for example, if we look at Hydra, which is shown in this example here, the Hydra produces a little bud, which is a clone of the original organism, and that bud falls off and establishes a new organism. That's asexual reproduction. There is no variation in genetic information from parent to offspring, because they are clones, they are identical. However, in sexual reproduction, which is where we're going to focus most of our discussion when we get into meiosis, in sexual reproduction we have 2 parents, and those 2 parents both contribute genetic information, and the unique combination of genetic information is what produces the unique offspring. One thing that's really interesting is when we look at humans, there are actually about 8 million different possible combinations of genes that you can have in each egg and in each sperm. And so if you were to look at the probability of producing the same genetic event twice, then you're actually looking at a very, very low number. So it's unlikely that two parents would produce the exact same combination of eggs and sperm, or genes in their eggs and sperm in an offspring. And so that's why all of your siblings look very, very different, but in some cases they can look similar. Okay, so let's talk about meiosis, because this is the process of creating these gamete cells which contain unique combinations of chromosomes and genes. It is the process by which gametes are made, and in meiosis it's going to be different from mitosis, because here we're going to see 2 rounds of cellular division. So here is our parent cell, and we do start off with diploid cells in meiosis. Okay, so we have a diploid cell, and that diploid cell is going to divide once to produce 2 cells, and then each of those cells is going to divide once to produce a total of 4 cells. And we end up with 4 haploid cells at the end of meiosis. So that's the first, that it has 2 rounds of cell division is the first thing. The second thing is that we do end up with haploid cells rather than diploid cells, and so we see a reduction in the chromosome number in meiosis. Now, one of the things that can happen in meiosis that is unique is that you can get something called crossing over that occurs. So in prophase 1, we see that our homologous chromosomes synapse, or they connect to each other. And so, if remember, we talked about homologous chromosomes means this would be a chromosome number 1 and this would be a chromosome number 1, but you got one from mom and one from dad. And so they carry the same type of information. Well, when they make a connection to each other, sometimes the genetic information kind of wraps around each other, the DNA breaks, and when the DNA repairs itself, it repairs itself so that the pieces of DNA have been swapped between the 2 chromosomes. And this is okay because they carry the same type of information. And so as long as that DNA is breaking in the same place, which it typically does, we don't see any problems as a result of that crossing over event because they carry the same type of information. And so you're really just swapping a blue eye gene for a brown eye gene on those chromosomes. But the reason that this is very important is because it increases the amount of variation that we see in our gametes, because it makes it possible now to have even more combinations of genes than you had before. So these are kind of explanations about what happens in the different stages of meiosis, and I prefer to look at pictures, and so do recognize that you can come back and look at this later, but let's go and look at the pictures first. So we separate the 2 rounds of meiosis into meiosis 1 and meiosis 2, and in meiosis 1 we see the same 4 stages that we saw in mitosis. We see prophase 1, metaphase 1, anaphase 1, and telophase 1. Keep in mind that if you do not put a number behind it, that your instructor may assume that you are talking about a stage of mitosis. So make sure that if you're talking about meiosis that you specify which stage, 1 or 2, you're actually talking about. So in prophase 1 of meiosis, we see many of the same things happen that we saw in mitosis. So if we review, in mitosis we saw that in prophase our chromosomes condense, so they become more tightly organized. Our mitotic spindle forms, and we see the same thing happening here, that the spindle fibers are forming. And the nuclear envelope breaks down, and we see the same thing in meiosis 1. So we see those same 3 basic phenomena happening in prophase 1 of meiosis 1 that we saw in mitosis. However, we also see those other 2 things that we just discussed, that our homologous chromosomes are going to synapse with each other, and so you can see here our homologous chromosomes have formed connections with each other called chiasma. And then as a result of that synapse, we can see crossing over occurring, which we see happening right here. Crossing over can occur between those homologous chromosomes to increase the amount of variation that we see in the gamete cells produced. And so that's prophase 1. Moving on to metaphase 1, If you recall from mitosis in metaphase, we saw that the chromosomes lined up at the metaphase plate, or that area along the center of the cell. And we see the same thing happening here, except if you remember in prophase 1, our homologous chromosomes formed connections with each other. Well, they're still connected, and so the homologous chromosomes in pairs line up at the metaphase plate in metaphase 1. And then in anaphase 1, we see that the homologous chromosomes are now going to separate. So instead of our sister chromatids being pulled apart, the homologous chromosomes are being pulled apart. And remember that all of this action, all of this movement of chromosomes and pulling apart of chromosomes is due to the connection of those spindle fibers to the kinetochores of our chromosomes that allows them to get lined up at the middle, and then also when those microtubules start to shorten in anaphase 1, that's what causes the chromosomes to be pulled apart. And then finally, in telophase 1, we're going to see that the spindle is broken down, that our nuclear envelope is reforming, and we might see a little bit of the chromosomes kind of decondensing or spreading back out. However, keep in mind that both of these cells that are going to result from telophase 1 are now going to go into another round of division, so they're not likely to get as spread out as you would see at the end of telophase and mitosis. Overlapping with telophase 1, we also see cytokinesis, which remember is division of the actual cell, the cytoplasm. And so we have our cleavage furrow here because this is an animal cell. And in the cleavage furrow, we'll see that that will eventually kind of pinch together and cause the cell to completely separate. And so now we'll see both of those cells move into meiosis 2. And again, here are the words, but let's look at the pictures. So we see in meiosis 2, we've got both of our daughter cells from meiosis 1. And in prophase 2, it's going to look very similar to mitosis. Our chromosomes condense back up, we see our spindle form, and we see the nuclear envelope break down. Okay? So we see those same things repeated over and over in prophase. In metaphase 2, this also looks very much like metaphase of mitosis, in that our individual chromosomes now line up at the middle of the cell, or the metaphase plate. And then in anaphase 2, this also looks very much like mitosis, in that the sister chromatids get pulled apart, and now these are called individual chromosomes, and they're going to be pulled to opposite ends of the cell by the centrosome. And then once they get to opposite ends of the cell, we see that the spindle breaks down, the nuclear envelope starts to reform, and now those chromosomes can decondense, kind of loosen back up and spread out. And overlapping with telophase 2, we see cytokinesis occurring, which is going to produce 4 haploid cells. So at the end of this process, we have 4 haploid cells. But what's really important for us to look at in terms of distinction is if we were to go back and look at meiosis 1, at the end of meiosis 1, we actually have 2 haploid cells. And if you go back and think about what does haploid really mean, it means that you have only one set of chromosomes in those cells. Well, if we look here in metaphase 1, here we have 2 sets of chromosomes, right? So they're paired up with each other. So if we look at this top row of chromosomes, that's one set, and the bottom row of chromosomes is one set. Well, those 2 sets of chromosomes get pulled into opposite cells, and so you've got one set of chromosomes that ends up in this new cell, and one set of chromosomes that ends up in this new cell. And so at the end of meiosis 1, we actually have 2 haploid cells, which then go through divisions to produce 4 haploid cells in total. All right, to wrap up and kind of finish up this whole unit, we're going to look at a quick comparison of mitosis and meiosis. We know that both of these are going to be types of nuclear division, okay, so that would be a similarity. They're both going to involve those same 4 stages, prophase, metaphase, anaphase, and telophase. However, if we start looking at differences, we see that in mitosis we only have 1 division, whereas in meiosis we have 2 divisions. In mitosis, our chromosome number stays the same from the parent cell to the daughter cells, whereas in meiosis, the chromosome number gets reduced. In mitosis, we have no crossing over. However, in meiosis, we do get synapsis of the homologous chromosomes, which allows crossing over to occur in prophase 1. In mitosis, we see single chromosomes lining up in metaphase and then sister chromatids separating in anaphase, whereas in meiosis 1, homologous chromosomes line up in metaphase 1, and then our sister chromatids stay attached in anaphase 1 because our homologous chromosomes are separating. So those are the big differences that we see between mitosis and meiosis. And keep in mind that mitosis is going to be a process that we see involved in growth of an organism. Or in some cases, for single-celled organisms, it may be involved in reproduction of that organism. But overall, if you think about what's happening, if you start as a single zygote cell, a fertilized egg, that zygote is going to go through a process of mitosis to produce 2 cells. And those 2 cells will go through a process of mitosis to produce a total of 4 cells, and so on and so on and so on. And so in order for you to grow into the millions of cells that you are, that are now differentiated into all different cell types, your body or your cells have to go through a tremendous amount of mitosis to get to that point. In order for small children to grow into adults, in order for you to repair, whenever you have some kind of a wound or problem, mitosis is going to be the thing that allows new cells to form to fix that. Meiosis is only going to occur in gametes, to produce gametes, so it's going to occur in your reproductive organs, in ovaries or testes, in order to create those eggs and sperm that we need as haploid cells to be able to join together and produce new zygotes. So it all kind of comes full circle, but you need to keep in mind, what those 2 processes are actually used for. Okay, that's it for chapter 13.